Welcome to this presentation about the hematology basics. A blood cell analyzer like the CYANHemato 380 can be used to perform one of the most often requested and informative diagnostic tests, the complete blood count. Before describing the system, let's discuss the basics to understand how the analyzer can produce a complete blood count. Today we will see the composition of the blood and which cell types are present, followed by the measurement principles. Some important notes on sample handling will be discussed, and finally we'll see the differences between our CYANHemato analyzers. Let's start with the blood composition. To understand the measurement principle of the analyzer, we need to understand the composition of the blood. For easy representation, let's have a look at a centrifuged sample. You will be able to distinguish three layers. First, you have the plasma. It contains salt, minerals, water, and proteins. The second layer contains the white blood cells and the platelets. It only makes up 1% of a blood sample. And finally, the red-colored layer contains the red blood cells. Each of these cells has a specific function in the blood. Let's have a look. The red blood cells are responsible for transporting oxygen from our lungs to all other organs in our body. In addition, these cells contain hemoglobin, which can also be quantified with hematology analyzers. Looking at the amount of these cell types in the blood, they are the most abundant cell type. Next, we have the platelets. Platelets play an important role in the coagulation or blood clotting, making sure bleeding stops. Looking at the size, these are the smallest cells in the blood. The white blood cells are important for our immune response. They defend us against pathogens, such as bacteria and viruses. They are the biggest blood cells and can be divided in 5 subtypes: monocytes, eosinophils, basophils, neutrophils, which are also called granulocytes due to the presence of granules inside of their membrane, and lastly, the lymphocytes. The monocytes, together with small eosinophils, are called the mid-sized cells. Granulocytes, they contain 3 different white blood cell types, and the lymphocytes. Now, the CYANHemato analyzers are 3 differential analyzers, because no distinction can be made between the 3 granulocytes. For this, you would need a 5-diff device, using more sophisticated and costly measurement components such as a laser. So the CYANHemato analyzers will report the mid-sized cells, granulocytes, and lymphocytes, without making a distinction between eosinophils, basophils, and neutrophils. Which properties are used to differentiate between all those blood cells? Let's have a look. There are 2 important aspects based on which the machine discriminates between the different cell types: the quantity of the cells and the size of the cells. Regarding the quantity of cells, we have to take into account that the amount of different cell types is different. In a blood sample, we will see that the red blood cells are more abundant than the platelets, which are more abundant than the white blood cells. This is easy to remember because it's logical. The red blood cells contain the compound that colors the blood red. If there were more white blood cells as red blood cells, the blood would be white. So, is the quantity of cells different in the hematology analyzers? No. The red blood cells are more abundant than the platelets, which are more abundant than the white blood cells. To be able to measure each cell type in correlation with each other, we will thus need to apply different dilutions. Dilutions to measure red blood cells and platelets, and a different dilution to measure the white blood cells, which are less abundant than the other ones. The next aspect, which is very important in measurements, is the size of the cells. When looking at the size of cells in a blood sample, we will see that the monocytes are bigger than the granulocytes, and these are bigger than the lymphocytes. Is this the same in hematology analyzers? No, surprisingly not. How is this possible? We will see that the granulocytes will now be bigger than the monocytes and the lymphocytes will remain the smallest white blood cells. In the analyzers, red blood cell and platelets are measured separately from white blood cells because otherwise there would be interference from red blood cells in the white blood cell counting. We will see that due to the addition of one specific reagent, the red blood cell and platelets will be destroyed and the white blood cells will all be reduced to their nucleus and granules. All of this caused by the lyse reagent. So red blood cells and platelets are destroyed, and now granulocytes will be bigger than monocytes, bigger than lymphocytes, because the membrane is destroyed and only the nucleus and the granules remain to be sized. So the lyse reagent has three functions. Hemolysis, which dissolves the membrane of red blood cells and platelets. This will ensure that there is no interference from red blood cells and platelets during the counting of the white blood cells. Secondly, this will allow hemoglobin quantification, because when the red blood cells are destroyed, the hemoglobin content is released and can be measured. The lyse will then react with hemoglobin to form a stable colored complex, which can be measured. And then thirdly, it allows white blood cell differentiation. Only nuclei and granules are preserved, and now the device can distinguish between lymphocytes, mid-sized cells, and granulocytes, the three diffs. Which measurement principles are used in the hematology analyzer to perform a complete blood count? Two measurement principles are applied in our analyzers. The first one being the impedance method or Coulter method, which counts and sizes cells by detecting and measuring changes in electrical impedance when a cell passes through a very small hole called an aperture. This principle allows to count the number of red blood cells, platelets, and white blood cells. The second method, which is spectrophotometry, is used only for the measurement of the hemoglobin concentration. It is a standard absorbance measurement of the red hemoglobin content in the sample. Afterwards, calculations and derivations from the histograms are used to obtain the results of the 20 reported parameters. Let's have a more detailed look at these principles. The impedance method is based on the fact that cells show great electrical resistance. In other words, they conduct almost no electricity. The cells to be counted are diluted until a single cell suspension in a fluid which conducts an electrical current, the diluent. This dilution of the sample occurs in the measurement chamber, which is flanked by an aperture and electrodes, negative and positive. By creating a vacuum, the cells are sucked one by one through this special narrow opening, the aperture. Cells will flow one by one from this side to this side. Flanking the aperture are two electrodes. They conduct electricity. When no cell is in the small aperture, electricity flows freely. However, when a cell is sucked through this aperture, the current is resisted. As such, there is a measurement of difference in tension. And the system will count the number of such events and also measures the pulse in the current, which is directly correlated to the volume, the size of the cell passing through. And so we will see that a big particle A, when it passes through the aperture, a big electric impulse will be recorded. When a small particle B passes the aperture, a smaller impulse will be recorded. So each event is counted as a cell, and the distribution of the cells is made by the electric impulse, the height of the electric impulse. So, by monitoring such pulses in electric current, the number of particles for a given volume of fluid can be counted. The size of the electric current change is related to the size of the particle, enabling a particle size distribution on a histogram. But how can the system differentiate between the three white blood cell types, lymphocytes, mid-sized cell, and granulocytes? If nothing would be done, you see that the size of the lymphocytes is overlapping with the red blood cells. The system can use a separate timing or chamber to measure the fractions, but also the lyse reagent is mixed with the blood used for the white blood cell counting. As such, we will obtain histograms for each cell type separately. White blood cells, red blood cells, and platelets. The mid-sized portion contains monocytes together with some eosinophils. And the granulocytes contains many neutrophils, some eosinophils, and basophils. The second method the CYANHemato uses is a standard absorbance reading, required from the measurement of the hemoglobin concentration. Hemoglobin has a red color, but you can only measure it when it's free in solution. Therefore, we add lyse to the cells, and this will destroy the red blood cells and set the hemoglobin free. Then, a green LED that emits light at 540 nanometer is located around measurement chamber is turned on. The free hemoglobin in the sample in the measurement chamber will absorb this light, and the remaining emitted light is detected by a photodetector. So the system calculates how much is absorbed and thereby how much hemoglobin is in the sample. Proper hematology sample preparation and storage ensures accurate, complete blood count. Some important notes are discussed in this chapter. To start with, the blood sample needs to be collected in specific tubes with EDTA anticoagulant. This prevents the platelets from clumping. Make sure that the sample tube is filled until the indicated line, because underfilling causes an excess of EDTA and will change the cell morphology. The cells will shrink, which affects the results. Regarding the storage and transfer, the collected samples can be kept maximum 8 hours between 15 to 30 degrees or maximum 24 hours between 2 and 8 degrees. When transferring to another lab, do this in a box with cold packs, but place the samples in between towels or bubble wrap to avoid freezing. Freezing causes hemolysis and affects the results negatively. Before measurement, it is important to have let the sample stabilize at least 30 minutes after collection at room temperature. This is needed for the effect of the anticoagulant to be optimal. Then homogenize by rolling in the hands for 20 to 30 seconds, followed by 20 slow inversions. Never shake the sample and do not use a mechanical shaker. This will damage the cells. We will end this presentation with an overview of our CYANHemato analyzers and get to know how the measurement process allows to obtain results for the 20 hematology parameters. Both our CYANHemato analyzers use the impedance and spectrophotometric principle, but differ in design and the amount of tests per hour that can be measured. The CYANHemato can do 60 tests per hour, while the CYANHemato 380 can do 80 tests per hour. This is due to the fact that the CYANHemato only has one measurement chamber and needs to process first the red blood cell and platelet counting, followed by white blood cell counting, and then the hemoglobin spectrophotometry. The CYANHemato 380 has two measurement chambers and can do the red blood cell platelet as well as the white blood cell counting simultaneously and is thus faster. The stages of the measuring process are as follows. First, the needle goes into the sample tube and aspirates a small volume of blood. Then, this blood is diluted in the white blood cell chamber. The needle now aspirates a small portion of blood of this diluted blood sample and moves toward the red blood cell chamber for a second dilution in the red blood cell chamber. Meanwhile, in the white blood cell chamber, lyse is added to the mixture. Next, the counting of the cells happens at the same time in both chambers. So in one, the red blood cell and platelets are counted, in the other one, the white blood cells. After this, hemoglobin measurement is also performed. After this, there is a washing and blank of the hemoglobin, and then both chambers are washed. So in the end, we obtain a complete blood count. By combining the Coulter and the absorbance measurement, the CYANHemato can measure and derive in total 20 parameters. Total count of white blood cells, red blood cells, and platelets. there is white blood cell differentiation, which gives us a total count of lymphocytes, monocytes, and granulocytes. We obtain hemoglobin content, the mean content and the mean concentration, the volume of red blood cells, hematocrit, platelets, and thrombocrit. And finally, we also get the distribution width of red blood cells, platelets, and two research parameters, the platelet large cell ratio and the platelet large cell count, 20 hematology parameters.